Okay, I'm Dr. Rocío Díaz de la Garza. I work here at Tec Monterrey. I teach undergrads and grad students, and I am a researcher in plant molecular and cellular biology.
I'm Dr. Guy Cardinal. I'm the director of the Center for Active Biotechnology at the Monterrey campus of Tec de Monterrey. I've been working in the area of agricultural biotechnology for approximately 30 years. I teach graduate students and undergraduate students here. Biosafety is obviously the approach to evaluating risk associated with biological experiments for us anyway. And I have been the chairman of the Biosafety Committee in one of the companies for whom I worked. I had about a 20-year career in industrial agricultural biotechnology. And then, of course, when I was a professor at Arizona State University, we had an institutional biosafety committee there, and all experiments that are proposed using genetically engineered approaches to doing things have to be evaluated by the institutional biosafety committee if you are looking for funding from any federal source. In order to get funding from a federal source in the United States, you must follow the guidelines that are established by the National Institutes of Health. So primarily for me, every time we wrote a grant proposal, we would have to then write a document for the Institutional Biosafety Committee outlining what our approaches were and how we were in line with guidelines from the National Institutes of Health. And we're trying to establish a very similar sort of situation here, particularly with regard to genetically engineered plants, but this will also overlap into other programs and projects that involve the use of recombinant organisms.
My experience working in the lab under biosafety rules began in the United States at the University of Florida, specifically working generating transgenic organisms, both plants and prokaryotes. And so I know how the system works in the US, and now that I'm here in Mexico, I know which are the rules here in Mexico. There is an institute that is CBOHEM, is the one that at the national level is in charge of setting those rules. The rules are set, and they are a mixture or a combination of rules that apply in the US and those that apply in Europe, mostly regarding to transgenic plants. The rules for a lab usually are pretty much the same everywhere, and in Mexico they are set. The thing is, we need to implement them, and the government is working on that. We here, because many of the professors that work here have worked in other labs, we usually, the biosafety rules that we use and apply with our students are based on general terms such as, for example, transgenics need to be sterilized before throwing it away, or things like that. So here in Tec de Monterrey, we have a set of rules that everybody knows and have to follow. Those are written and they are delivered to any student, undergrad, grad student, or professor that is working here in the facilities. On the other hand, we have another lab that with animal facilities, in which we have a biosafety committee that follow and check all of the protocols that before they are conducted. On the other hand, CONACYT, we need to assure CONACYT in every grant proposal that we have all of the instruments in place, and we follow all of the biosafety rules in order to get grants as well.
I reviewed all of the protocols that the iGEM 1 team is using, in which they describe in great detail the approaches to using a variety of very common laboratory chemicals and techniques, including running DNA gels, running protein gels, isolating DNA, isolating proteins, and so forth. They identified a variety of different chemicals and their potential dangers, and also how they would handle them specific to particular dangers that may be inherent to their use. All of the protocols are outlined in reasonable detail and how things would be dealt with, involving the use of personal protective equipment, hoods, biosafety hoods, and so forth. And so the protocol is also perfectly reasonable to me, and I think what they plan to do with regard to addressing issues associated with basic laboratory safety are more than adequate. I think their approach to doing things is to use general laboratory safety protocols, and there are no biological experiments that they are proposing, as far as I can tell, that have any extraordinary risks beyond what we would normally find in the laboratory where people are doing genetically engineered or genetic engineering bacteria or yeasts or other organisms of that kind.
Thank you.
From my perspective, the primary thing is much like medicine: first, do no harm. So the objective is whatever it is that we're anticipating doing, or whatever it is that anybody is anticipating doing, we need to make sure that we know the most that we can possibly know about potential outcomes. And part of what biosafety entails is evaluating risk. So I would argue that we do risk assessments or risk analyses every day. We talked about that in the bioethics class. So we're always doing risk analyses. We're not even aware of it. But in relation to biotech, we have to do theoretically formal risk analyses where we actually consider what it is we're trying to do and what happens if a bacterial organism or a plant gets out of the lab and gets out into the environment. What's the risk of that? What's the potential for that happening? And if it does happen, what's the possible outcome? So from a biosafety perspective then, we're not going to allow experiments that have a high element of risk. Example, this is an example we used last week. Would not allow someone to try to make a multi-drug resistant bacillus anthracis, right? Because we already know that can be used as a bioweapon. It has been. It's highly toxic. If you inhale it or it gets into wounds, it produces a toxin. The toxin's very toxic to mammals and can kill you fairly easily if you're not treated within a short period of time. How are you going to be treated? Doxycycline or Cipro are the primary antibiotics. We made a multi-drug resistant bacillus anthracis resistant to those drugs. You couldn't prevent anybody from getting ill. Now the good news with anthrax is that if you get it, you can't give it to me. It's not communicable. But you can imagine if you did that with, say, Yersinia pestis, which is the plague bacteria. And if you get it in your lungs, it is communicable. I can give it to you by breathing on you. So now we would have a huge problem. One of the difficulties is that it's impossible for us to completely identify all possible outcomes from the experiment. So you might remember we talked about the mousepox experiment that the Australian scientists did, where they fused interleukin-4, I think it was, to mousepox. And the idea was to try to stimulate immune uptake of the virus. They were trying to look for ways to build better vaccines against smallpox or other similar viruses, and instead they almost completely wiped out the immune system of the mice, and almost all the mice died. So that was not a good outcome, but it was an unanticipated outcome. Those are the scary things. When you're trying to do something for a positive purpose and you end up with an unanticipated outcome, it's almost impossible to, without any question, determine it. But I think we have a pretty good idea. There's some things you just don't do, and we try to evaluate those things that we're doing to make sure we don't do any of the things that we just don't do. Okay, so
with regard to the experiments the iGEM team is doing, they're pretty basic sort of experiment. You're looking for very specific things. You're trying to address an allergy-related issue, for example. Allergies are incredibly important. If we could find ways to treat allergies or cure allergies, it would be very beneficial. So, is there no risk associated with it? I can't really say that because there's virtually nothing for which there's no risk. It's the probability of the risk becoming an unfortunate event. That's the critical parameter, I think. We have to make a judgment based on evaluating approaches and data and so that's sort of